Sanger's method of DNA sequencing was based on Arthur Comberg's earlier work on DNA replication. Remember that a new DNA strand is synthesized using existing strand as a template. To illustrate the technique, let's use polythymine DNA as an example. The 5' carbon of incoming deoxynucleotide DNTP is joined to the 3' carbon at the end of the chain. Hydroxyl groups in each position form a ester linkage with the central phosphate. In this way, the nucleotide chain elongates. The key to Sanger's sequencing method is the peculiar chemistry of dideoxynucleotides. Like deoxynucleotide, DIDNTP is incorporated into a chain forming a phosphodiester linkage at its 5' end. However, the DIDNTP lacks a 3-hydroxyl group necessary to form the linkage with the incoming nucleotide. So the addition of DIDNTP alters the elongation. To sequence a DNA, four separate reactions are necessary. One to provide the sequence information about each of the nucleotides. Each reaction contains a template DNA, a short primer about 20 nucleotides, DNA polymerase, and 4 DNTPs, one radioactively labeled. One type of DIDNTP is added to each. Let's focus on adenine terminator reaction. First, the DNA is denatured into single strands at near boiling temperature. At high temperature, the kinetic motion of DNA molecule disturbs the weak hydrogen bonds that complementary DNA strands together. When the temperature is lowered, the primer binds to its complementary sequence in the template DNA. Kronberg's earlier work has showed that a primer is needed for a DNA polymerase to start replication. The DNA polymerase makes no distinction between dNTPs and dIdNTPs. Each time a dIdNTP is incorporated, in the case dIdATP, synthesis is terminated and the DNA strand of a discrete size is generated. In this sequencing example, dIdATP, which is in purple in color, has terminated the reaction. The dATP happens to be a radioactive tracer, but this has no effect on elongation. Because billions of DNA molecules are present, elongation reaction can be terminated at any adenine position. This results in collection of DNA strands of different lengths. The same is true for other three terminator reactions. Each reaction is then loaded into a separate lane of polyacrylamide gel containing urea, which prevents the DNA strand from renaturing during electrophoresis. Ionized phosphates give the DNA molecules a negative charge, so DNA migrates towards the positive pole of an electric field. The movement of DNA molecule through the polyacrylamide matrix is size dependent. A blue dye that also migrates to the positive pole is added to the samples to track the progress of DNA in the sequencing gel. The dye runs just little faster than the smaller fragments of DNA. Over the course of electrophoresis, shorter DNA molecules will move further down the gel than larger ones. Millions of terminated molecules of same size will migrate to the same place in the gel. After the electrophoresis, the gel is sandwiched against a X-ray film. The radioactive adenine in the synthesized DNA emits beta particles that expose the film, making a record of the position of DNA bands in the gel. The sequencing gel is then read from the bottom to top. The sequence bands in various terminated lanes give the sequence of nucleotides in the template DNA.